Hello, welcome to the lecture on consciousness. So, in the earlier segment of this lecture, we talked about the link between stimulus and awareness. In fact, throughout the lecture we never talked about how the experience, the conscious experience that the stimulus creates. We only talked about the responses in the brain, right, which part responds to vision, which part responds to auditory stimuli and things like that. We never really consulted the subject, the experiencer, right, we never brought the experiencer into the picture. So, in this lecture we began to do that in the first segment, but in the first segment we only talked about this kind of a variable relationship between the stimulus and the experience that it creates. So, for example, we had given some examples of color perception. So, color we know from physics is rigorously defined by the frequency of the light, the frequency or wavelength of the light. We have seen examples in the previous lecture where the color perception varies a lot, and from person to person it can depend upon the lighting, it can depend upon the context, and it can depend upon whether you are seeing the original image or it is after image and so on and so forth. So, there is a lot of variability that comes in when you introduce consciousness. So, we got a bit of feel of that in the first segment. So, this segment we now look at neural substrates. So, what happens in the brain when this kind of variations in consciousness or conscious interpretations of stimuli occur. Let us begin with a curious experiment called the rubber hand experiment. So, in this experiment the subject sits on a table, both the hands are placed on the table like this, palm down, and so in this case this lady her left hand is actually behind that white screen, and what you are seeing with what looks like her left hand, which is right in front of the white screen, is actually a toy hand, a dummy hand. It is just a rubber hand. Only thing is it is designed so that her actual hand, her actual right hand, and her this kind of a dummy model of the left hand both look very similar, and the screen is placed such that the lady cannot see her actual left hand, which is behind the screen. And you see the experiment is actually touching with both his hands, with his left hand is touching the rubber hand which is placed in front of the woman, and with his right hand is touching the actual left hand of that subject, the woman. So, now the experimenter actually starts stroking the rubber hand which she can see, which the subject can see, and also her actual left hand which she cannot see, but she can feel. So, her brain is actually receiving two stimuli, the visual feedback from the rubber hand which looks like her left hand, and as the person stroked the hand she can see that, okay, and then the somatosensory or touch feedback from her actual left hand which is behind the screen, which is also touched by the same person and stroked. Now, this stroking now has a pattern. Now, he is stroking two hands, one rubber hand, one real hand. He makes sure that the stroking is done in the same way, the rhythm or the pattern of stroking is identical with both these hands, with the rubber hand and the real hand. So, as this happens like for a minute or so, right, and then the subject starts feeling that the rubber hand is actually her own hand. She starts identifying herself with that rubber hand, and she starts like owning it up. She starts she starts imagining that it is part of her body. And now, in one experiment which is slightly dramatic, they actually took a hammer and tried to bang on the rubber hand, and the subject jumped up in alarm thinking that she is going to get hurt, but it is only the rubber hand. So, the identification is so deep. So, what is happening here? Can we rationalize this and explain what is going on? So, the subject's brain is receiving input, visual input from the rubber hand and the touch input from her actual hand, and both these inputs are actually similar, the patterns are similar. So, now there is this theory that brain tries to identify objects and based on their attributes using what is called feature binding. So, look at this simple question. So, I am looking at this red cricket ball, its color is red and it is round in shape, right. So, both are visual properties. So, now imagine there is a region in the brain, the visual cortex of the person, so let us say A, and then there is some firing pattern in response to the roundness of this object, and the firing of these neurons represent round, the property round. There is another region B where the firing represents color, and this particular firing pattern represents red. So, now the brain should know that both these properties correspond to the same object, they belong to the same object. So, because any object is basically a combination of certain properties, the basic properties could be very small, not very large number, but different objects come with different combinations of properties in terms of color, shape and so on. So, how does the brain know that both these colors, both these properties represent the same object, they are associated with the same object. So, the feature binding by synchronous firing, this theory says that These two sets of neurons, one representing the shape property of the object, another representing the color property of the object, they actually fire in synchrony. So, therefore, if you look at these two pools of neurons in different parts of the brain and record from them, the fact that both are firing in sync tells you that both neurons are talking about the same object. So, basically the integrity of the object is interpreted by synchronizing in the time domain. So, basically what this theory says is, if you are looking at an object and now you are looking at different aspects of the object, right, and these different aspects they activate different parts of the brain. So, all those different activities represent the same object. So, therefore, they fire in sync. So, therefore, they will show synchronized or correlated neural activity. So, when different pools of neurons which respond to the same object or different properties of the same object, if they fire in synchrony or show correlated activity, then you perceive, the subject perceives that all those neurons are looking at the same object, are interpreting the same object. So, the oneness of the object is interpreted by the brain by translating it or coding it in terms of synchronized activity of neurons. So, correlation implies identity. So, just look at what is happening in the rubber hand experiment. So, the experiment is so kind of cunningly designed, so that the pattern of visual feedback, the movements of the finger of the experimenter, and the pattern of the somatosensory feedback or the stroking pattern of the experimenter on the real hand, both are identical. So, therefore, the corresponding neural response to both these stimuli, some in different parts of the brain, also are probably identical, right. So, therefore, the person is interpreting that both these stimuli are coming from the same object, her actual hand, so which was a mistake, because normally it does not happen, right. You never have a situation where, right, some replica of your hand is stroked and then right, and then your real hand is also stroked in the same way, that kind of thing, this is here it is experimentally created in a very cunning way, but that does not happen normally. So, that kind of a false correlations do not arise in real life experience, but in this they are creating that kind of a false correlation. That false correlation is creating a false identity, a false interpretation of identity. Okay, so, basically by tampering with the correlations among neural activities, you can tamper with the way we perceive the world, with the way we interpret objects in the world. Okay, so, now this is a theory, but but is there any experimental evidence for this theory? So, people have found that so this kind of a correlated activity particularly happens in a certain frequency band. So, whenever you are talking about synchronization, two neurons are firing, they are for them to be synchronized, they have to be oscillatory, they have to have some firing, they have to fire at some frequency, and then they sync at that frequency. Brain has many frequencies. I mean, if you take an EEG or electroencephalogram of a brain, you will find that the spectrum of the signal has multiple bands. So, some of the more important bands that you find in EEG are shown in this figure. So, there is a gamma band which goes from 31 to 100 hertz, then there is a beta band which goes from 16 to 30 hertz. The alpha band which is seen when you are in a quiet meditative state is between 8 to 15 hertz, the theta band from 4 to 7 hertz, and delta, which is generally seen in deep sleep, right, is from 0.1 to 3 hertz. So, now it turned out that there is some evidence that when you are looking at an object or multiple properties of the object are being responded to by different parts of the brain, neurons that respond to all these properties fire in synchrony in the gamma band. So, in fact, some of the pioneering work on this topic was done by a German lab headed by Engels and Gray and Singer and Koenig and others. So, what they performed an experiment with cats, cats visual cortex. So, they presented a moving bar of a given orientation to the cat, and we know that in the visual system you have this neurons which respond to oriented bars, and also some neurons respond to oriented bar moving in a given direction. So, they found they measured from neurons in different parts of the visual cortex of the cat which respond to the moving bar, and they found that all those neurons show synchronization in the gamma band. Okay, so, that was a major discovery. So, the kind of ideas that have been floating around, in fact, this idea was first proposed by Malsberg, Christoph Vander Malsberg, and that idea turned out to be true, and this kind of a feature binding by synchronization seems to be true, and there is this effort to discover more and more examples, more and more evidence for that kind of a feature binding. But that still only talks about object identity. How do we understand an object by combining and assembling its various properties? How do you understand that all the properties of the object are actually associated with the same object? How do you understand or encode object integrity? But that does not say anything about the object's perception. How do you see the object? How do you experience the object? It does not say anything about that. So, here is where there was a theoretical leap that was performed. It was proposed by Francis Crick in what is called the astonishing hypothesis. What Crick had suggested is that gamma band synchronization subserves not only object identity, which is what we have discussed, but also consciousness. So, that means unless the activity becomes synchronized in the gamma band, it does not enter consciousness, and whenever it gets synchronized, that is when you become aware of it. Okay, is there any evidence for that? So, this study by Lucia Meloni and colleagues, they presented a stimulus to a subject, and the thing is, when the subject is presented with a short stimulus, a brief stimulus, then you can find that the brain is responding to it. You can see it in EEG, you can see it in at single neuron level also. But if it is too brief, then subject shows no evidence of sensory awareness of that stimulus. But when you present it for a longer time, right, you will see that there is synchronized activity across a large spatial scales, right, in the brain. So, there is a long range transient synchronization in the EEG in the gamma range when the patient was conscious. And contrarily, right, in psychiatric conditions like schizophrenia, Parkinson's, autism, epilepsy, this kind of a synchronization is somewhat weaker. So, there are deficits in this kind of synchronization. So, the synchronization has some correlate with your conscious experience of the stimulus. And a very interesting set of experiments have been done by Benjamin Libet on what is called subjective timing. So, we talked about the fact that are you conscious or not when you present a stimulus, what does it take to become conscious. So, we just said that unless stimulus is presented for a sufficiently long time, you do not become conscious of it. So, that is a basic condition for you to become something conscious. But the other interesting question is, when do you become conscious? So, I present a stimulus to you, right, at exactly at what time do you become conscious of that, and here you have some very interesting results that emerged from the studies of Benjamin Libet. So, the thing is in this studies, so actually the cortex of the subject is patient is exposed, and they put electrodes on the cortex and electrically stimulate the cortex. So, they initially play with so the current is passed into the cortex, and the current pulses, right, the current amplitude and the current frequency and the pulse duration, these are varied, and this search for what is the minimum condition, right, that what is minimal stimulus that it takes to produce consciousness in the subject. So, what they found is, even if your current intensity is slightly lesser, in the present same train of pulses, sequence of pulses for a longer duration, right, the person becomes conscious. But thing is, there is a minimum that you need, minimum current level that you need for the person to be conscious, and at that minimum you have to present it for sufficient duration. So, this minimal stimulus, right, present to the cortex, which produces the conscious experience in the subject, this Benjamin Libet calls it the liminal one, and at this minimum current intensity you have to present the stimulus for about 0.5 seconds, right, for the subject to be conscious of the stimulus. Okay, so thing is, so if you so what is interesting is, when you stimulate the cortex, you need to stimulate it for nearly half a second before you find that the subject feels that stimulus, the effect of the stimulus. But what is interesting is, what happens when you actually give a peripheral stimulus. So, suppose you are stimulating somatosensory cortex, right, when you stimulate it directly in the cortex, then you feel that somebody is touching some part of the brain, depending upon where you are stimulating. So, let us say I am stimulating the kind of a middle finger part of the somatosensory cortex, and when I stimulate there, subject will feel that his or her middle finger is being touched. Now, in another experiment, I actually touch the middle finger, and then I ask, when does the subject become conscious of the stimulus. So, if you directly stimulate the cortex, right, it takes about half a second for the person to be conscious of it, right, and then cortex is right there. I mean, your brain, your mind, you would think is right here inside the brain somewhere, and you would think that the hand is far off from your brain. So, it is much farther from your mind, and therefore, if you stimulate the hand, you should take longer for it to enter your conscious awareness, but the results are different. Okay, let us see what happens in actual experiment. So, here we have defined 5 quantities. C indicates cortex and S indicates stimulus or skin stimulus given superficially, and object represents objective world. So, the time, so Tc subscript object means, so the time at which the stimulation is given to the cortex. So, that is in the objective world, and that you can measure with your clocks and stuff. Then Tc sub is the time at which the cortex stimulus is felt in the subjective world, So the gap between the TC obj and TC sub, that means time at which you begin your stimulation, time at which the subject feels that he has felt the stimulation, the effect of the cortical stimulation is about half a millisecond. This is what you can see in the the middle line, you see that middle line. Now there are 3 more time quantities, Ts obj and Ts sub, that is the time at which the skin is stimulated in the objective world and the time at which the subject feels that the stimulus is received in the subjective world, that because that is something the subject alone knows. And then there is a third thing, which is the Ts sub, but expected. When do you think the skin stimulus would reach the subjective the subject the subjective conscious awareness of the person. So normally what you would think is when the cortical stimulation is given, the gap between the TC obj and TC sub is about half a second. So you would think that same thing gets carried over even when you stimulate the finger or the skin. Actually you would think that it will take even longer, but quite paradoxically the Ts sub is actually very close to the Ts obj. That means the time at which they feel the stimulus is received is actually much quicker than what is expected. So that means if you summarize this result, when you stimulate the brain directly, stimulate the cortex directly, it takes much longer for the stimulus to enter the consciousness, that is you, the conscious self. But when you stimulate the skin, it is entering the conscious self much quicker, actually it takes only a few tens of milliseconds, compare that with 500 milliseconds for the cortical stimulation. So first of all, one big question that arises in interpreting these kinds of experiments is, how do you measure something like subjective time? For example, I have a thought, I have a thought that I want to go and drink some water, and that thought occurs at certain time. Now how do I time it? I cannot compare the thought with some clock, and how do you do that? So what in the experiment what they have done is, the subject is shown a dial on which there is a dot moving round and round, that he has received that stimulus, he has felt that stimulus. So at that point he looks at this dial and sees where the dot is on the screen, on the dial, and then he remembers that location, because since the dot is not moving very fast, the subject can remember the location of the dot with reasonable accuracy, and that is given to the experimenter later, and then they average this over many trials, and that is how you time the subjective event. Okay, these are subjective timing experiments. Okay, so therefore when you stimulate the finger or the skin, right, it actually reaches the consciousness much quicker than when you stimulate the cortex. Benjamin Libet has a very interesting interpretation of why this is happening. He calls this a subjective referral backwards in time. So basically what he is saying is, when brain knows that when you give a stimulus, for it to reach the conscious self, it is anyway going to take 500 milliseconds or half a second. This happens all the time. So if I want to more accurately judge when the stimulus has occurred, may as well introduce a correction all the time, so that I am better off, right, in determining exactly when the event has happened. So your conscious self or your brain is making this correction all the time, right, when you get stimulus from outside, through the regular natural channels of sensory stimulus. But then a brain does not normally get stimulated directly in the cortex. That is a unnatural thing which has begun only recently. So brain does not do this correction when you do cortical stimulation, but when you give stimulation peripherally in the skin, it introduces kind of a correction. So the conclusions by Libet are, the cortical activity due to sensory stimulus must persist for a minimum duration to produce conscious sensation, and he calls it the neuronal adequacy. So you need to have activity in the neuron for a minimum amount of time, and you get to generate minimum activity in the brain, right, for that to enter your conscious awareness. And once the adequacy is achieved, the subjective timing of the experience is referred backwards in time, because brain knows through evolution there is always this delay between the external stimulus coming from the external world and the subjective event that it produces. Okay, so that is some interesting experiments about subjective aspects of sensory stimulus. Right, in the last segment we talked about how the stimulus is experienced. Until now in this segment we talked about when the stimulus is experienced, and we saw some aberrations, very interesting aberrations, and now we will see similar aberrations which will shatter our kind of intuitive beliefs about the motor side or how actions are performed. So we all, even if you look at a textbook on neurology or neurophysiology, so they divide actions into voluntary and involuntary movements. For example, if you step on a pin and then you involuntarily by reflex, right, you withdraw your foot, that is called involuntary. You don't, there is no conscious involvement in that kind of a response, whereas you are sitting quietly and then suddenly you get up and go and drink some tea or something like that. So, that is a completely internally driven, a voluntary movement. Okay. So, so that is what is called generally free will, and there is all the debates in the philosophical circles whether there is free will and all that. But if you just go by the popular understanding that there is such a thing called free will, right. And how does free will work? So, normally what you would think is your will or your your motivation or intention to move starts first, and then somehow that gives rise to certain activity in the brain, neural activity in the brain, and depending upon which part of the body you want to move. Suppose you want to move your right hand, so then we know that that is controlled by the left brain, left motor cortex, and then somehow activity appears there like magic almost, and then from there it goes to the spinal cord and activates your hand. So, how exactly does free will work? Now, we would assume that even when there is sensory stimulus, between sensory stimulus and the motor response, there is some kind of a subject, a self, a kind of a conscious agency, an agent, which inserts himself or herself between the sensory input and the motor output, right. And then this is what determines whether you want to act or how you want to act and things like that. Okay, but that is what you would assume, but there are patients in which this kind of interaction, this kind of a gating, this kind of insertion of the conscious self does not seem to be happening. Okay. So, it is not simply a question of decision making, because in the subjects studied by Francois Le Hermite, right, these people are slaves to the sensory world. So, if you show them for example a pen, right, they will just pick it up and start writing on any surface. If you show them a comb, they will just pick it up and start combing their hair, even though there is no need for it at that moment, they will just start doing it. Or if you give them a toothbrush, they will start brushing themselves immediately. Okay. So, if you show them a bed, maybe they will just go and lie down on it, right, even if it is totally inappropriate for that context. I mean, there are a lot of people and it is maybe daytime or something. So, they are completely, their actions are completely driven by the external stimuli. There is no self or agency which inserts itself, right, between the sensory input and the motor output and decides whether to act, and if so what exactly should be the action. So, you see this little cartoon picture in the bottom. We have discussed in our one of the early lectures on organization of nervous system, that between the sensory input and the motor output there is this very high region of the cortex, the prefrontal cortex, right, which performs decisions and all that, and it is here there is a damage in these subjects, and so they are not able to they become slaves of the external stimuli. In fact, this condition is called environment dependency syndrome. Now, okay, but that does not happen normally. So, normally in normal subjects we have the feeling that we have a will, and then that, right, if you look at the normal account of how we think we move on our own, it goes something like this, right. The will first arises in the mind, you have an intention, you have a thought, okay, I want to do something, and then actually if you look at the functional imaging scans of people who are engaged in voluntary movement, that is the subjects are asked to say lift their finger on their own at a self appointed time, there is no stimulus which is telling them okay do it, they decide on their own and make a simple movement. And when that happens, people found that the first activity is seen in the supplementary motor area, and that happens bilaterally. The supplementary motor area is located more medially, and this activity happens bilaterally. After that, the activity then slowly progresses to the M1 to the contralateral side. So, what begins as a bilateral activity becomes unilateral, becomes actually contralateral, goes to the opposite side of the part of the body which is moved, and then the activity flows down towards spinal cord, towards to the hand, and then the movement occurs. So, the normal sequence of events that you would imagine is the first the conscious event, the event of will, right, when you want to the intent to move, then the brain's activity, whether it is SMA or primary motor or whatever, some kind of some activity, some event happening in the brain, then the actual movement. But actually this simplistic belief that we all carry with us all the time, right, and that is what our sense of self is based on, this simple belief seemed to be shattered by this elegant experiment by Gray Walton. So, in this experiment the subjects had electrodes implanted in the SMA, so they can stimulate the SMA, and then the subjects can actually press a button, and then in front of them there is a screen, and there is also there is a slide projector which changes the slide on the screen. So, what they were told is by pressing the button they can change the slide on the slide projector. That is what they were told, but actually it is a lie. There is no connection between the button and the slide projector. So, what is actually happening is they are picking up electrical activity in the brain from the SMA, and that activity, whenever they see the activity there, that means there is intention has begun, and they use this activity to control a lever and a machine, which will activate the slide changer and then slide moves forward. Okay, so they are kind of in sense preempting what the subject is wanting to do. So the subject keeps pressing, and then he sees the slide moving. So normally, if you think that the sense of will is born first, the subject will be conscious about the intent first, and then would observe this. So would observe the slide moving. But actually, what happened was the side projector seems to be anticipating their intentions, and moved even before the subjects have intended, not even before pressing. The subjects just barely feel, right. Actually, even before they had the intention, the slide is already preempting their intention and moving. Okay. So that means your SMA activity begins first, and that activity is driving this hidden machinery, which is moving the slide projector, right, and making it change. And then this person simply presses the button, right, after the activity has started in the brain, but that has no connection to the real change in slide. So they feel that actually it is as though the slide changer knows their mind and responding even before they actually think of changing the slides. Okay, so the subjects became aware of their intent a bit late, and that is what it looks like. Let us look at it more closely. So normally, whenever we do things on our own by willed action, by what is called voluntary activity, you would there are three events which take place: the will, the arising of the will event, and the motor cortical activity in the brain, and then the actual movement. So the normal sequence in which these three events will be thought to occur, that is what we would believe, is will first, motor cortical activity later, and then the actual movement. But so whereas Benjamin Libet has done an experiment to show that this sequence is not quite correct. Okay, to understand the experiment, we have to mention that there is this thing called readiness potential. So like I said, whenever you move things, right, on your own voluntarily, the brain's activity that is observed when you perform voluntary action is quite different from the brain's activity when you perform, say, stimulus driven action. So suppose somebody is asked you to move your hand, and then immediately when you see the stimulus you move the finger, right, that activity, the brain's activity that you see, say, in SMA and motor cortex, is quite different from what you would see when you are moving your hand at your own self appointed moment. So what exactly happens? So thing is, in case of voluntary movement, when you decide to move on your own, brain starts preparing for that event well in advance, almost like close to a second, or more than half a second before the event. So in this graph, you see some graphs on the right side. So you see the third graph from the bottom, right, that is mid parietal. So this is close to the midline. So here you see that this activity is building up almost once more than 0.5 seconds before 0. 0 is the time when the movement has occurred. Okay, so it is well in advance, the movement is the activity is building up in the brain. So this is called motor preparation. That means brain is preparing for movement, getting ready to move, okay, all getting charged up to move, and this takes a long time in case of voluntary movement. Whereas if you are just responding to a stimuli, that happens much quicker. You do not need that happens maybe 100 milliseconds or even less than that. Okay, so this is called the Bereitschafts potential in German, and a familiar English word for that is readiness potential. Basically, it conveys that the brain is getting ready to move, right, in the form of this kind of a voltage. So now Benjamin Libet's experiment looks at this kind of measurements from brains when the subjects are engaged in voluntary movement. So like I said, the BP or RP, as the potential is called, represents C, the motor cortical activity. Then you can measure the movement by hooking up the person with electrodes, so that the electrodes pick up the muscle activity, the muscular activity, and that kind of measurement is called electromyogram. Myo denotes muscle. So that measures electrical activity in the muscle, and that event represents M, which is actual movement. Now there is a third event, which is willed action, that is when does the will event occur. So there are three events that you have to measure and compare them. So again, like before, the willed action event or will event is measured by using this kind of a moving dot arrangement. So the normal order, the intuitive order which you would expect is will first, motor cortical activity later, and then actual movement. But what Libet's experiment has shown is motor cortical activity first and then will, and then actual movement. In fact, you can see that that is pretty much what is happening in Gray Waters experiment. So, there the motor cortical activity started first, and the subjects felt that they are already preempted. I mean, they were still thinking about it, already the slide has moved. Only thing is, in Gray Waters experiment they have not actually measured the time at which the will occurs, but that is what they have done in the Benjamin Libet experiments. But basically the qualitative result is same in both cases. The time of will event happens later, after the motor cortical activity. So, what does it mean? It basically tears down that the idea, that simple belief on which our lives are based, that we are the bosses. So, there are certain things which you do by coercion, somebody asks you to do and you do it, but there are certain things that you do on your own. But even in those things where apparently you are doing completely on your own voluntarily, right, it is as though brain decides to move and do it, and then you are informed, the conscious self is informed by the way. So, it is like brain initiates the action all by itself and is just courteous enough to inform you by the way, right, about the proceedings that were initiated right under the nose of the conscious self. It is like brain decides to move your hand and then it says, by the by, right, I am moving your hand, I just want to inform you, right, but you have no control over it. So, that is a bit scary, but this is what the experiment seems to indicate. So, Gerald Edelman, a Nobel laureate, right, who has dealt with the question of consciousness, so talks about that there are two realms, the C and the C prime. C is the world of conscious experience and its contents, what you feel, what you experience, when you experience and how intensely do you experience, all this, all the contents of the subjective world, your emotions and moods and all that. I mean, it is difficult to quantify all that, it is difficult to measure them, put numbers on them, but they exist, I mean, we know that, right. And then there is the objective world of stimuli, you have lights and pressures and sounds and all these things that you can measure and quantify, right, measure with your experimental equipment. Now, the question is, the big question in consciousness research is, how are these two worlds related, right. If I give a stimulus and I know what stimulus it is, I show a color blue, which I know is definitely blue because the wavelength that comes from it can be measured by a device and I know that wavelength corresponds to blue. But when you show it to a person in certain conditions, the person might think it is, I don't know, green or something like that. So there can be a discrepancy between what is out there in the world and what is experienced in there, right, in your conscious self. So, linking these two and working out the relationships and all the variability and richness of this relationship is a big challenge in the consciousness research domain right now. So, Dahane and co-workers have done this interesting masking studies. So, where they were shown some words visually. So, they were shown very briefly and then after that the word is masked. So, they won't know what the word is because it is presented for a brief time. So, we know that there is a thing called neuronal adequacy, but unless stimulus is presented for a very long time, sufficiently long time, it does not enter your awareness. So, in this case the words are presented for a short time and then they are masked by some other stimulus which covers it up. So, when they do that, they have found that there is activity in the primary visual cortex. So, brain is responding to it, but the subject is not aware of it. Okay. So, you see already the kind of discrepancy between brain's responses and the subject's responses. But when the words are presented without mask for a longer time, right, then subjects are conscious of it. But there is a very interesting difference between the kind of brain's responses that are seen in these both cases. In the first case, when the masking stimuli is presented, the activity was confined to the primary visual cortex, so basic lower sensory areas. But when the activity, when the word was unmasked and presented for a longer time, the subject became conscious, and there is much widespread activity in the visual, parietal and even frontal areas. That means, can we say that unless the higher areas of the brain's hierarchy, these are parietal on the input side and the prefrontal on the motor or executive side, unless these areas are involved, right, in the response to stimulus, in the brain's responses to stimulus, the subject does not get any awareness of it, right. Brain responds, you can measure those responses using all sorts of experimental equipment, but the subject is not aware of that. So, based on these kinds of studies, Bernard Bars has proposed this global workspace theory. Basically what he proposes is that there are large specialized networks, right, subconscious by themselves might be working together to achieve awareness through synchronization. There is a very interesting theory proposed by Giulio Tonini and Gerald Edelman. So, what they propose is that there is this tightly connected network of brain areas in the higher levels of the hierarchy, right, again between parietal and prefrontal and so on. They form a very tightly coupled network and they call it the dynamic core. And the dynamics, this dynamics of this core must be sufficiently complex. And what we mean by complex, I mean they are able to define that mathematically using ideas from information theory. So, from computer science information theory, and these theories have found some evidence in a lot of data we have from EEG, from people who are in different states of consciousness. So, for example, in the case of a subject undergoing a seizure, there is huge activity in the brain, but subject is actually unconscious while that is happening. So, you can show that the activity in this case has actually, it has high amplitude, but has a low complexity of dynamics according to theories of Thorney and Edelman. Then there are also physical theories of consciousness. So, for example, Susan Pocket and John Joe McFadden have proposed that the electromagnetic field generated by neural activity, that is substrate for consciousness, and then synchronized activity in this EM field, right, is a correlate for consciousness. So, it is not just a signal, but also looking at the EM field, and then some other question is, do you have to modify the Maxwell equations as you study in electrical engineering to study the field propagation in the brain. So, these are all open questions. And one more question that arises is, when you start looking at physical theories of brain, is that in consciousness experiments you see a lot of distortions of space and time, right. Your event in the external world is something, the time duration, and internal world is something else, right, and then your measurement of space is something in the external world and something else in the internal world. So, normally we take these distortions as a kind of aberration. We take the physical world as the standard, right, as a absolute reference, and with respect to that we take the response of the brain, we take the interpretation of the subjective self as some kind of an aberration, some kind of a variable thing which has to be constantly checked and validated and calibrated with respect to what we know as the time and space in physics. But maybe in future, right, these aberrations cannot be taken as simply aberrations, but you have to deal with it. So, then you have to have even a physical theory of consciousness which can integrate these aberrations that are found in the conscious experience domain research, and also that aberration and the kind of theories of space and time as are formulated in physics. So, maybe in future we will develop a more comprehensive theory of consciousness which kind of includes our understanding of space and time as it occurs in physics. So, right now the situation is there are a wide variety of brilliant theories, and in fact there are a lot of theories that originate from quantum mechanics, and there is people talk about quantum gravity, microtubules in neurons and how they are related to consciousness and so on. But there is no consensus in all these theories, and there are different pockets of theories, and which is the situation with most questions in the brain, and lots of theories without any consensus. So, it is obviously a very hard problem, and a lot more progress has to happen. But if that progress can happen, and we can really understand consciousness from a scientific point of view, I mean not like how the religious people have been discussing it or how psychologists have been talking about, but purely from a scientific point of view by the standards of empirical and objective science, if you can have a theory of consciousness in future, I mean that will probably make a major impact, right, on human society and civilization. Thank you.